So hi guys, this is quiz number 3, and the first question asks to draw don't know how to pronounce it, a phylogenetic tree of 3 domains of life. Label the phylogenetic tree with class distinguishing each domain from the next. Additionally, identify one monophyletic group and one paraphyletic group on the tree. Just wanted you guys to see this. Now I'm going to close it. So, here's my tree. Kind of suck at art. So, the phylogenetic tree has 3 domains. This one is my bacteria, archaea, and eukarya. I think the other question asks, so identify the clades. So on my bacteria, this mark right here is my clade, and that's the cyanobacteria and the gram-positive bacteria. And for my eukarya, it is the animals, fungi, and plants. And I know that it asks something about a monophyletic group. So the monophyletic group that I'm going to choose will be the animals. And of the animals, I'm going to say dogs because I think that was the example given. And the paraphyletic, I'm sorry if I'm pronouncing that wrong, will be the different breeds of the dogs. I also Googled further into this and seeing that, you know, some fish could be monophyletic, and then the different species of the fish, like shellfish and things like that, could be the paraphyletic groups. So that's question number 1. Sorry, I have to look at the question again. And it closed. I'm just going to show you. Opening it again so that I can get my questions. Screenshot my questions. No, that isn't it. Sorry. Sorry. So my questions, because I screenshot it. So again, here's my questions. So number 2 says list and describe ways in which microbes can be classified based on their phenotype. So microbes can be classified by their shape, their size, and also their structure. But you can only do this because microbes are so small with viewing them under the microscope. So phenotype. So with the phenotype, by identifying them by their shape, would be like if it's a coccyx, which is the circles, or if it's spherical, and that's considered, it's kind of what it is. Spherical is the circle, sorry, which is cocci. Or if it's rod shaped, the size, of course, I think prokaryotes are smaller than the eukaryotes size microbes. So I think prokaryotes, it says that it has like 70S, and eukaryotes has 80S. And then its structure. So I would say its structure because we do stain these. So so identifying the microbes phenotype by its structure and its staining abilities, I would say like the gram-negative and positive, which one stains purple and the other one stains like pink. So you can identify microbes phenotype by its shape, size, structure. I guess I'm kind of putting structure, or with its staining characteristics. Okay. So the next question asks, list and describe 2 ways in which microbes can be classified on their genotype. Closing this, guys. I'll put it over here. So 2 ways that microbes can be classified by genotype. It's really interesting because we discussed it in the last lecture. Is DNA sequencing. It's where they can take parts of the fragments and or this DNA sequence and I guess identify what this particular microbe is. The other one is RNA sequencing, and per the lecture, this one is a little bit better to use for identifying or figuring out the genotype of a microbe because RNA seems to be a little bit more stable than DNA because RNA can't really undergo too many mutations without it going haywire. So the other question asks, imagine you are a microbiologist working in a research lab and given the task to differentiate between 2 strands of viruses. What steps will you take to complete this task? So, what steps would I take to complete this task? Per the lecture, again, it talked about doing biochemical testing. And with a type test, you can get the different strands or types of viruses or bacteria that's present. And I think from there, the gel electrophoresis will be probably the next best step, so that you can possibly see what DNA is lagging or the heaviest or whatever. And if not the gel DNA, I'm going to go back to the old lecture again, would be the DNA fragment test. And that right there will just kind of show me what DNA is present within this virus or this strand. And then from there, the CRISPR technique. So now, since you have this very specific strand isolated, you can possibly come up with the genome for treatment. Again, I'm sorry, I didn't what the question asked. So how would I go about verifying, I think, the 2 strands? So I would do a biotest to kind of single out the strand, and then do electrophoresis, a gel electrophoresis to kind of narrow down some of the sequence in that virus or bacteria. Well, the virus, because that's what it asks. And then my last step would possibly be the CRISPR, where I take that part of the genome and study it more and possibly come up with treatment. And I know the lecturer spoke about the different strands of influenza, but like influenza, Ebola has a bunch of different strands too. So that will be my plan of attack.